This lecture will be about the nucleus, and I'm Dr. Hadwa Ali, Assistant Professor of Histology, MTI University, Faculty of Medicine. When discussing the nucleus, this description will be about the interphase nucleus. The meaning of interphase, it means the nucleus of the non-dividing cell. Interphase nucleus, it is the nucleus of non-dividing cell. When we want to discuss the nucleus, there are certain items that are important when describing it. First, the number. Some cells have only one nucleus, and in this case, they are called mononucleated. Mono means only one nucleus. For example, this is a nerve cell, and we can see here that it has only one nucleus. Also, some cells have two nuclei, and in this case we describe these cells as being binucleated. This cells has two nuclei. Or there may be more than two nuclei, so in this case we describe the cell as being multinucleated, more than two nuclei. Some cells in our body, or the only non-nucleated cell in our body, is the red blood cells. The red blood cells has no nucleus, so it is not regarding the site of the nucleus. The nucleus might be apical in position. Apical, it means it is in the apical part of the cell. Or it might be central, in the central part of the cell. Or it might be eccentric. Eccentric means it is present, or the nucleus is present midway between the center and the cell membrane. Also, the nucleus might be peripheral in position. It means it is close to the cell membrane. Regarding the shape of the nucleus, there are different shapes. For example, the shape of the nucleus might be flattened, or it might be rod shape, or it might be oval in shape, it might be rounded. The other shapes of the nucleus might be kidney shaped, or it might be bilobed, as in this cell. Bilobed, it means it is formed of two parts connected by a strand of chromatin, and what is meant by chromatin, we are going to describe this term later on. Or the nucleus also might be multi-lobed, formed of different or 3 to 4 segments, connected also by a chromatin strand. Also, the nucleus might be in the form of C-shaped or horse band or horseshoe. Also, it might be taking the form of this regarding the appearance, lobulated nucleus. The appearance of the nucleus might be of two forms. The nucleus might be darkly stained, and in this case we call it dark phase nucleus or inactive nucleus. As we see here, this nucleus is darkly stained. We cannot see any details inside this nucleus, and this is a photo of an inactive or dark phase nuclei. Also, the nucleus might be open phase or vesicular. Open phase nucleus, we can see the nucleolus inside it, and we can see the details of the nucleus. As you see here, this is the nucleus, and we can see the nucleolus. So we describe this type of nucleus as an open phase or vesicular nucleus. Regarding the components of the nucleus, there are 4 components of the nucleus. The nucleus is surrounded by a membrane, and as you see here, this membrane is formed of an outer part or outer nuclear membrane and an inner nuclear membrane. And if you look to this membrane, it is interrupted at certain sites by nuclear pores. The nucleus also contains nucleolus, chromatin strands, and nuclear sap. Nuclear sap is the material which is present between the chromatin and the nucleolus. So these are the 4 components of the nucleus. We're going first to start studying the description or the structure of the nuclear envelope or the nuclear membrane, first by light microscope. By light microscope here, we can see that the nuclear membrane is formed of a single basophilic line, or appear as a single basophilic line. Why it appears as a single basophilic line? It appears basophilic for 2 reasons. First, as you can see here, the outer nuclear membrane is continuous with the rough endoplasmic reticulum, which is studded by ribosome. And the inner nuclear membrane is associated with chromatin, and this chromatin is formed of DNA. the ribosomes on the rough endoplasmic reticulum is formed of RNA. So these are the causes
The structure of
make
the
them or which make
nuclear
this
membrane
membrane
by
appear as a
electron
basophilic
microscope.
line.
By electron microscope, more details could be seen, and we can see that the nuclear membrane is formed of an outer membrane and inner nuclear membrane. The outer nuclear membrane, as you can see here in this diagram, is continuous with the cisternae of rough endoplasmic reticulum, which its surface is studded by ribosomes. The inner nuclear membrane is related to a structure called chromatin. And also we can see in this diagram that the inner nuclear membrane is supported by a group of nuclear or intermediate filaments which form what is known as fibrous lamina or nuclear lamines. These nuclear lamines
The nuclear envelope
is
is interrupted
supporting
at
the inner
certain
nuclear
sites
membrane
by
structures called nuclear pores. And at this site or at the site of the nuclear pores, we can see that the outer and inner nuclear membrane are continuous with each. What is the function of these nuclear pores? These nuclear pores act as site of exchange between the cytoplasm and the nucleoplasm. The second structure in the nucleus is the chromatin. What is chromatin? Chromatin is a DNA strand which is wrapped around histone proteins. شريط ال DNA or each DNA molecule is a very long strand، بيبقى شريط طويل، فالنيوكليوس حجمها صغير، لا تمكن أو لا يمكنها استيعاب هذا الطول لكل مولكيول من ال DNA. عشان كده كل مولكيول من ال DNA لازم يلف حوالين بروتينز اسمهم هيستون بروتينز. This wrapping occurs at different degrees, and the maximum condensation of chromatin strand occurs during the cell division, and they are called chromosomes. What is the function of the chromatin? As I said, chromatin is a DNA strand wrapped around the chromatin. They direct and guide protein synthesis by formation of three carries the genetic RNAs, and also the chromatin carries the genetic information which will be transmitted to the next generations by a highly coiled, and they are genetically inactive. Also, they cannot undergo transcription. They are called heterochromatin. Other parts of the chromatin strand are more or less extended. In this case, they are genetically active and they can be transcribed, and they are called euchromatin. في بعض الأجزاء من الكروماتين ستراند تبقى لفة على بعضها جدا وبتبقى كوندنسد جدا، وبالتالي لا يمكنها أن تؤدي إلى مثلا formation of RNA، مش هتعمل RNA ولا بيبقى عليها جينز اكتف تتنقل للسكند جنريشنز او للاذر جنريشنز. لكن في اجزاء تانية من شريط الكروماتين ستراند بتبقى نوعا ما مفروده، وبالتالي ممكن من خلالها ان احنا نعمل عمليه ترانسكريبشن ونصنع انواع مختلفه من الار ان اي، وفي نفس الوقت بتبقى جينيتيكالي اكتف يعني عليها جينز، والجينز دي تبقى ترانسميتد للسكند جنريشن عن طريق عمليه الدبليكيشن. The electron and light microscope. What is the difference between hetero and euchromatin? First, by the H&E staining or by light microscope, we can see that heterochromatin will appear as a basophilic granules، حبيبات لونها ازرق بتبقى جوه نيوكليوس، هي دي اماكن الهيتيروكروماتين. while euchromatin cannot be seen، بما ان الهيتيروكروماتين بتبقى highly condensed وبتبقى لفه على بعضها، فانا ممكن اشوفها داخل النيوكليوس على شكل حبيبات زرقاء. اما الايوكروماتين فلا يمكن رؤيتها. by electron microscope, heterochromatin will appear as an electron dense masses، هتظهر على شكل اجزاء لونها اسود في اماكن مختلفه. اما بالنسبه للايوكروماتين it will appear as an electron lucent areas or electron lucent color. What are the different shapes for heterochromatin by electron microscope? There are peripheral heterochromatin which are present in relation to the inner nuclear membrane. Also we have chromatin islands. These are parts of the heterochromatin scattered in the nucleoplasm. Also we have nucleolus associated chromatin. They are the parts of the heterochromatin associated with the nucleolus. So can you complete this comparison between EU and heterochromatin? Then let's come to discuss the remaining two components of the nucleus, which are the nucleolus and the nuclear matrix or the nuclear sap. Let's come to the structure of the nucleolus. Nucleolus by light microscope appear as a rounded or single basophilic or a basophilic mass within the nucleus. Why it appear basophilic? Try to answer this question. Also, the nucleolus will be seen more, or it is prominent in the nucleus of a protein secreting cell. Why? If you come to the function of the nucleolus, nucleolus is responsible for formation of ribosomes. So this is the cause that it is more prominent in the protein secreting cells. The number of the nucleoli by electron microscope. The nucleolus, as you can see, is formed of two parts, darkly stained parts and lightly stained parts. The last structure of the present in the nucleus is the nuclear sap or the nuclear matrix. This nuclear sap or nuclear matrix is the colloid solution that fills the space between the spaces in the nucleus. What is meant by the nucleolus and cell cycle is different types that occurs in the cell during division and during the resting phase, which is called interphase, or in other words, it is the life cycle of the cell. So it is formed of two phases, a resting phase which is called interphase, and it is a long phase as you can see here. All of this is called interphase, and a dividing phase or a division phase which is either mitosis or meiosis according to the type of cell. Thank you.